ರತ್ನಾಕೋ ಶೋರೂಮ್ಗೆ ಸ್ವಾಗತ. ನಮ್ಮ ಕೆಲಸದ ಸಮಯ
Hi, Madam Josna, Madam. Good morning.
Good morning, Andy. How are you doing?
Fine, fine. Thank you. How are you, Madam?
I'm sorry, it's 11:30 here, so
Yes.
I might yawn in between, in the presentation. So don't mind.
We are actually disturbing you.
Not at all. Actually, it's not a problem at all. I'm happy to, present.
Yeah, yeah. So within 5 minutes, we will start, Madam.
So who are the audience? I are like
Audience, students, scientists, teachers,
okay,
and university officers. All we have 42 institutes under YSR Horticulture University. So, polytechnic colleges, degree colleges, race stations, and KVKs. So, all will connect.
okay, sounds good. So can you hear me well?
Yeah, yeah.
Okay.
All right. Perfect.
అలాగే ఓకే అండి ఓకే సరే అండి థ్యాంక్ యూ గుడ్ మార్నింగ్ అండి.
మేడం గుడ్ మార్నింగ్ మేడం. మేడం గుడ్ మార్నింగ్.
గుడ్ మార్నింగ్ మేడం. గుడ్ మార్నింగ్
జ్యోత్స్నా దేవి.
Yeah, good morning.
డాక్టర్ పద్మావతమ్మ is calling you.
Namaste,
నమస్తే మేడం.
So please don't call me madam. Jyotsna is fine.
I'll call Jyotsna.
Jyotsna. Madam Padmavatam madam, shall we start?
అందరు వచ్చారా?
జాయిన్ అవుతున్నారు.
ఓకే, మళ్ళీ మేడం కూడా లేట్ అవుతుంది కదా, స్టార్ట్ చేయండి.
11:30 అక్కడ 12.
నైట్
11:30 మేడం.
ఇప్పుడైతే స్టార్ట్ చేయండి ప్లీజ్.
Good morning. Welcome to Dr. YSR Horticulture University, Venkatramanagudem, West Godavari District, Andhra Pradesh, for the Global Connect series in collaboration with the University of Wisconsin, Madison, USA. The topic is molecular physiological aspects of cranberry and gooseberry, and Madam will concentrate mainly on cranberry. And respected Honorable Vice Chancellor, Dr. T. Janakiram, sir, and really with his support and encouragement, we have started Global Connect series in university. This is the 8th series, and already we have completed 7 series. Every month, one we are conducting, and so this is the brainchild of our Honorable Vice Chancellor, Dr. T. Janakiram, sir. And welcome. And today guest speaker Dr. Jyotsna Devi Neura, and the cranberry molecular physiologist, USDA, United States Department of Agriculture Research Service, and Department of Horticulture, University of Wisconsin, Madison, USA. and respected Dean of Horticulture, Dr. Padmavatama Garu, PG Studies, Dean PG Studies, Dr. Surya Kumar Garu, Director of Extension, Dr. Karunasri Garu, Registrar, Dr. B. Srinivasul Garu, Controller, Madam Shashikala Garu, Estate Officer, Mallikarjun Rao Garu, and the Dean of Student Affairs, Dr. Salome Sunita Garu, Controller of Exams, Dr. Venkataswamy Garu, all associate deans, all zonal research heads, and all heads of KVKs and horticulture research stations, and the principals of polytechnic colleges, students, teachers, scientists, ladies and gentlemen. And with this, good morning to all. And before going to details, I welcome to our Honorable Vice Chancellor and Dr. Jyotsna Mura. And before going to details, I will just briefly introduce Dr. Jyotsna Devi Mura, United States Department of Agriculture, and ARS Agriculture Research Scientist, Cranberry Physiologist. Dr. Jyotsna Mura is presently working as a research scientist with USDA, ARS, and Wisconsin, Madison. And first, her role is cranberry improvement, productivity, quality by exploring the fundamental mechanisms underlying the physiological response of cranberry to water, nutrients, and environmental factors. And Madam is Dr. Jyotsna is from Andhra Pradesh, from Kadapa district, and already Madam Jyotsna completed BSc and MSc from different Telangana and Andhra, and PhD from ICRISAT, Hyderabad. And after that, Madam got, she was awarded a fellowship from the US Agency for International Development and to conduct research at the University of Florida. After joining PhD, Madam worked at Florida, North Carolina State University, and with several international organizations, and USDA, ARS in Maryland. And she also worked in cotton, soybean, peanut, chickpea, common beans, cocoa, gooseberry, blueberries, and cranberries. And presently, she is working as a plant physiologist and molecular biology, biochemistry, and bioinformatics. And mainly, she is working on the improvement of the yield and production, productivity, and quality parameters in cranberry. And Dr. Jyotsna is having two children, two are sons, and at 7 and 11 years age. And husband, Dr. Srinivas Behra, also from working in United States Food Drug Administration, USFDA, and present designation is research molecular biologist, and she published 70 articles, 40 peer-reviewed articles published in national and international journals, and one book also published. And she has guided one master and two PhD students, and she has one visiting student from China also. And mainly, Dr. Jyotsna is working as a physiologist, molecular physiologist at the USDA. And welcome, Madam. Welcome to Dr. YSR Horticulture University and in the Global Connect series. And so welcome.
Yeah, thank you. Yeah, thank you very much, Dr. Dhananjay Rao Garu. So, and good morning, everyone. And there are so many people, I cannot name each and everyone. So thank you very much. First of all, I would like to thank you all for inviting me to present my research work. And especially, I would like to thank Dr. Dhananjay Rao. So I know he gave some introduction about me. I would like to tell about myself, few things. So my name is Joshna, as you all of you know, and I am from Andhra Pradesh. My family is originally from Kadapa, but we settled in different places like Guntur, Vizag. Um, so yes, I studied in different places, and my PhD is from ICRISAT. While pursuing my PhD, I got a fellowship from USAID, which is US Agency for International Development, and I moved to University of Florida. So since then, I'm working in the US only. I am a molecular biologist with the US Department of Agriculture. It's a federal agency, just like ICAR, IARA in India. But my lab is housed in the Department of Horticulture, UW-Madison. That's why I'm also an assistant professor with the UW-Madison. So today, I can share my screen, right?
Yes, yes. Yes, ma'am.
Yeah, I am doing it. So basically, I am a physiologist and also a molecular biologist, primarily working with cranberry. So I joined this position 2 years back during COVID time. US Department of Agriculture has different branches. I work with like almost 22 branches. I work with agricultural research system. Our mission is to work closely with the growers and identify their problems and find solutions to their problems at the national and global level. So today I'm going to present integration of multi-omics in cranberry and goji berry. So my work is primarily with the cranberry. But I'm going to briefly talk about goji berry too. So this is the overview of my presentation. First, I would like to talk about the cranberries because nobody grows cranberries in India. So we don't know anything about cranberries. So I just want to give brief introduction about cranberry history, facts, and domestication. And I would like to talk about omics and how we applied omics in cranberry molecular physiology research. That is my lab. And goji berries. I'm going to talk briefly about goji berries and how we applied omics in goji berries. So first of all, cranberry history, facts, and domestication. So cranberry is a unique crop. So it is one of the 3 native fruits that is cultivated in the North American continent. So the other 2 are blueberries and Concord grapes. So it is important part of the Thanksgiving meals in the US. Thanksgiving is a big deal in US. So along with turkey, people eat cranberries either as a dry fruit or fresh fruit and also as a sauce. Cranberry sauce is very popular. So these native cranberry plants, they survive under acidic soils. They love acidic soils and they require very low nutrients. And most of the agronomically important crops, they cannot survive under these conditions where cranberry grows. So cranberry belongs to the family Ericaceae, and there are 2 types. You can see Vaccinium macrocarpon, which are cultivated cranberries, and this Vaccinium oxycoccus and Vaccinium microcarpon, they both are wild ones. So cranberries prefer acidic soils with a pH range of 4 to 5, and they like the soils moist and also well drained. So, and also they like soils with rich organic matter. So technically they grow in peat soils. So main yield component is definitely fruit. And cranberry growers love fruit quality, like fruit size. They like bigger fruit and they like red color. And fruit firmness. Fruit firmness is very important for cranberries because most of the people eat dry cranberries. So they want, when they cut the fruit and try the fruit, when they process everything, they wanted it to be firm. So these are the traits that cranberry growers look for when they breed cranberries. So native, I want to talk about why cranberry got this name. Native Americans, that means the people, native tribes here, they usually call with different names. So when the European settlers came to US, one of the tribe, Algonquins, they shared these cranberries with Europeans. So they named them as cranberry because you can see the fruit, so the flower of the cranberry resembles the head and bill of the European crane. So that's why Europeans started calling them, European settlers started calling them as cranberry. And over the years, the name is shortened to cranberry, and they are still calling it as cranberry. So cranberries are cultivated in beds. So you can see like it's a huge bed here. We call them as beds only. Beds are box. Each bed is usually like 4 to 5 acres. And the beds are, you can see like they are a little lower than ground level because growers usually flood them a lot for different purposes, like for insect management, to kill the insects, and pest management, and also to protect from winters. There are different purposes they flood, and also for harvest. So that's why they have ditches and they are below the ground level. And also they use sprinkler irrigation to water them. So this is how it looks. This is very small plant. So maybe 6 to 8 inch tall. And these are the runners. And you can see like these vertical stems, we call them as uprights. And it has very fine fibrous root system here. So these are perennial vines and it has trailing habits. So you can see like these uprights are called like either vegetative uprights or reproductive uprights. Apparently reproductive uprights have fruits, and cranberries have biennial bearings. Biennial bearing means they yield a lot in 1 year and the next year they yield very less. So, and then the following year they yield more. So cranberries do have biennial bearing, but over the period with breeding and all, like with genetics, breeding and everything, now they have very less, all these hybrids have very less biennial bearing. So cranberries are popular in the US, first one thing, and they're also unique because of their harvest, water harvest. So people love to see their harvest actually, like harvesting the cranberries. Um, so I want to give some introduction about this harvest. Like these are in 1800s. So you can see people used to harvest them with the hands. Then in 1900s, they started doing mechanical harvesting. So it's called dry picking. You can see like they used to use these machines to pick up the berries. And now later on, they started doing water harvest. So cranberry fruits have locules. It means air pockets, these 4 air pockets that make them to float on the water. So that's the reason cranberries like to do water harvest. It is much much effort and efficient than mechanical harvest. So this is the one machine that you use for water harvesting. What they do is basically they harvest the beds. Like I said, like the beds are lower than the ground level. And then they use this shaker to shake the stems of the cranberries, and then the fruits float on top of the water. when they float on the top of the water, they rag them. So they bring them together at one place. And then they use, this is a video, I can play video for you. They collect them into the trucks. You can see. So these berries will picked up from here, and there are two vans. This van one van separates the trash, and one van collects the berries. So that's how they harvest them. Sorry, and they load them into the trucks. So and then they process the fruits. So these cranberries weren't always cultivated. So the Native Americans did not cultivate them. So they just gather all wild berries, and they use them for different purposes, like for medicine, as a food, and also food preservatives and dyes. So later, these cranberries, when the European settlers came to US, they started using them for different purposes, like as a tea to calm the nerves, and also for bladder, stomach, and liver disorders, and also for diabetes and healing wounds and other conditions. Today cranberry is most commonly promoted for urinary tract infections. So this is like, even if you go to the doctor, they suggest you to eat cranberries for urinary UTIs. So they are rich in vitamins, especially vitamin C, and cranberry has naturally contains this benzoic acid, which is known as food preservative. So many people use in foods also. So these are the different products of cranberries. You can see like they make them as fresh cranberries, dried cranberries. They make they make juices, and they use them in the chocolates and ice creams and the hand soaps. And they also make wines out of cranberries. This is cranberry wine. And there are different products of cranberries nowadays. So there are so many uses and so many products. Yeah, they use in like teeth whitening also. And for immune development. So like in between the presentation, I put some important facts about cranberries. Like during Second World War, American troops consumed almost 500,000 kilos of dried cranberries every year, because it has vitamin C and it provides immunity. That's why they consumed those many cranberries. So these are the US growing regions of the cranberry. So then they don't grow in southern parts because cranberry requires certain chilling hours. So that's why like most of the cranberry growing regions are northern US states. So you can see like mostly 5 states grow almost all of the country's supplies of the cranberries. And Wisconsin, where I am working, so that produces more than half of all cranberries of the United States. So you can see Wisconsin is the top producer of cranberries, followed by Massachusetts. Quebec is in Canada, and some British Columbia is also in Canada. Some in New Jersey, then New Jersey, Oregon, and Chile is another country. So there are different places that produces cranberry, but most of the northern states in the United States produces these cranberries. So, and after that, like they get exported to different countries. So you can see like these are the top 10 US export markets for cranberries, like Europe, UK, Mexico, China, Canada, Australia, Chile, and different Asian countries. So now we have more than 70 varieties of cranberries, and some growers, they do cultivate native varieties. They have been cultivating since 1800s. So, and I think like 50 years back, they produced first generation of hybrids, and Stevens is the most popular cultivar now. And recently they produced super hybrids with all like genetics and breeding technologies. So you can see how they improved the size of the cranberry with all these technologies. So these are the natives, and these are the first generation cranberry fruits. So you can see like these are the super hybrids now. So they tremendously increase the size of the fruit. So, and also they reduce the biennial bearing. So that's an advantage of breeding. Um, Now let's move to the topic omics. So what is omics? I think you all know it's a field of biology that ends with the omics, like phenomics, genomics, transcriptomics, proteomics, and ionomics. So phenomics is nothing but morphological and physiological characterization of crops, and genomics, most of you know like DNA sequencing, genomic mapping, and genetic profiling. So transcriptomics is nothing but RNA sequencing and studying the gene expression profiles and transcriptional regulation in the plants. Proteomics, as the name says, it is protein identification, quantification, and protein-protein interaction studies. Ionomics is measuring mineral nutrients and trace elements in the plant tissues. So why these are important? So all this integration of the omic technologies, they are applicable in the crop improvement. It's nothing but system biology. You can see like top-down approaches like phenotype to genotype. You can use all these omics technologies and identify different things, and also bottom-up approaches. At the same time, you can identify the genotype and you can make a phenotype. So using all the technologies. So technically, these are important for dissecting important developmental clues and gene characterization. They can predict the phenotypic responses and metabolic pathways in the crops. And also you can understand how cultivars like varieties respond to environmental interactions. So, and also we can produce abiotic or biotic stress tolerant phenotypes using all these omic technologies in the crop breeding. So how we are applying these omics in cranberry molecular physiology research. So as a USDA scientist, we usually work closely with the growers. Our national program leaders, they go and talk to the grower association presidents, and they identify the priorities like problems, what they have with the particular crop. And then they bring back to the scientists like me, and we discuss like what needs to be done. And they make it as a priority for us. So some of the things that I'm doing for the growers also. For example, like this phenomics. Um, Always growers want to find the basic information of all the cultivars, like whatever new cultivars produced, they want to have all the information. That's why I'm using phenomics here to get the basic information on new cultivar phenological development and the growing degree days needed for each cultivar to move from one developmental stage to another developmental stage. So I'm using ionomics here to study the soil pH problems in cranberry. There are some cranberry growers who are getting high pH because cranberry loves acidic soils. High pH is a big problem for them. So they want me to study that. So I'm using those ionomics. And also I'm trying to identify abiotic stress tolerance mechanisms in cranberries by using molecular and by studying molecular and physiological mechanisms. So here I'm using metagenomics and transcriptomics to study cranberry soils and roots. and identify beneficial microbes to use as biofertilizers. So let's come to the phenomics. So for phenomics, I'm developing a camera for cranberry growers to study cranberry phenological stages. So as I showed you, cranberry crop is very small. It's only 6 to 8 inches. So we need a high definition camera if I want to study each growing stage and when they are moving from one growing stage to another growing stage. I have to take pictures daily. So I need to have really good phenotypic camera. So these cameras cost around $1,000 or $1,500. We don't want to spend that much money. So that is the reason I started developing these cran cams for cranberries. So here technically I'm using a technology, Raspberry Pi technology. I think nowadays even high school kids are using these Raspberry Pis, to develop, like to do small projects like identifying motion sensor and ringing the bell, those kind of things. So these Raspberry Pis are very small compute. You can see this is the Raspberry Pi. These are card-sized computers. Runs on Linux-based program. So it has, like you can see, like USB ports, so many USB ports. You can connect different sensors like cameras, temperature sensor, light sensors, and humidity sensors to them, and you can collect the data. Here I connected a small camera here, and you can also connect different cameras, also infrared cameras and visible, like multispectral cameras or different things. So these cameras, these Raspberry Pis have Wi-Fi controllers. So it is very easy to collect the data. You can implement cloud technology with it. You can sit at home and collect the data. And these Raspberry Pis comes with open source software. So you don't need to buy any software to run the program. So it's very easy. So it's a cost-effective strategy for the phenotyping. So these are the Raspberry Pi cameras that we developed for cranberries. You can see, like here, Raspberry Pi computer, and I have microcontrollers and cameras. So, and we installed them in the field. So we connected them with the solar panels so that we don't need to connect to the power outlet, and also run whole growing season. And these are packed inside a weatherproof case. So even if rain comes or anything comes, like they will be fine. So we install them in the fields, and we capture images every half an hour, every day for 5 months to study different varieties of cranberries, how they are moving from one growing season to another one growth stage to another growth stage. So my PhD student, Dani, is developing those cameras. You can see, like how we can use these cameras is technically, as I said, to track developmental stages of different cranberry varieties. And also we are studying how these varieties are responding to different environmental factors, like because you can attach sensors to the cameras, you can always study like how they are responding to different environmental factors or radiation, and also from location to location, because I'm studying in Wisconsin and also I'm studying Massachusetts, another state where the temperatures are different from Wisconsin, and also in New Jersey. So how this data can be used? So this data can be used for selection of different cultivars, like some cultivars develop color early, some cultivars flower early. So those kind of things, you can always select if the breeder wants or if the grower wants particular cultivar for that particular location, they can always select based on this data. And also we want to develop phenological models, like growing degree day models and different models for growers to use. And also when each cultivar is moving, from one stage to another stage, you can always optimize your management, like when to irrigate, when to fertilize, when to spray pesticides, and when to harvest. These plannings can be done easily. So as I said, like we are collecting the images. So we are collecting thousands and thousands of images daily from different locations and from different varieties. So we cannot look at each image and say, hey, this cultivar is moving from this stage to this stage on that particular day. So we don't want to do that. That's why my student is developing these mission algorithms. So to process the images. So as I said, like we are using different platforms to process those images. And this is one of this, like my student is also developing some tools to count the cranberries and to identify the colors, which one is more color, which one is less color, and also identifying the color of leaves, like based on leaf color, we can always say which one is has more nitrogen, which one has less nitrogen leaves. So you can see like she developed a tool like where you can put fruits and it can count easily. So this is YOLO model that we developed. So these are the different cultivars. We studied different cultivars, but here I'm showing 3 cultivars. So we calculated based on this image data and based on that mission algorithms, we calculated growing degree days for each cultivars to move one growing stage to another growing stage. You can see like after dormancy, all these 3 cultivars started at the same growing degree days. But when you see this cultivar Malika Queen required more growing degree days to blush. Blush means to turn into red color than Crimson Queen. So this one is the early cultivar based on this data. And also Crimson Queen moves into blossom very early than any other cultivar. So usually growers fertilize with nitrogen at blossom stage. This always helps them to plan giving like fertilizer way ahead than Malika Queen and the Stevens cultivars. So that's way like these tools are useful for the growers. So next one is the Inomix. So in using Inomix, we studied how the soil pH impact cranberry fruit size. You can see like, as I said, like cranberry loves acidic soils. So when they are put under like high pH, because some growers are, even though with soil amendments giving granular sulfur or everything, so they still get high pH because of water, irrigating water. So you can see like these fruits, which are grown under optimum pH for cranberries, and these are high pH for cranberries. We found 40 to 60% of the reduction in the fruit size. So growers wanted me to identify the problem. Then we used this, we measured all nutrients, leaf nutrients, soil nutrients, fruit and stem nutrients, and to see which nutrients these are lacking, what are causing reduction in the leaf fruit size. So here you can see this bar is technically optimum pH bar. So optimum pH line. So the bars above this optimum pH lines shows the percentage of increase in the nutrients, and bars below this line, optimum pH line, shows percentage of decrease. You can see like except for the calcium and magnesium, which most of the high pH soils will have. So all other nutrients are low in high pH soil, high pH leaves and stems. So we found like reduction in mostly like micronutrients. That is the case. When the pH increases, you will find less micronutrients. And also we found less nitrogen, potassium, and phosphorus. So overall, we found a significant drop in micronutrients in leaf and stem, and also in fruit actually. And then we measured, because these cranberry growers are giving a lot of nutrients. They are fertilizing the soil. So we want to make sure whether these soils have these nutrients or not, where that fertilization is going. So these soils are rich in everything, but plants are not able to uptake these nutrients because of pH. So with the Inomics data, we found that soils have enough nutrients, but plants were not able to uptake any nutrients because of high pH. And they lack macronutrients like phosphorus, nitrogen, and also micronutrients at high soil pH, which might be responsible for the reduction in the fruit size. So our solution was like, our next research is to see if foliar sprays can help with these cranberry growers. So we are still working on it. We don't have data yet on using foliar sprays. So next one, I used transcriptomics. Transcriptomics, as I said, study of the gene expressions, identifying the genes. So we want to use these transcriptomic tools to identify cranberry cold tolerance genes. You can see like northern all US northern states have severe winters. So one of the strategies that cranberry growers employ is like they flood. You can see like they flood whole bed, and they wait until it forms an ice. So you can see like it forms an ice layer, and they draw water out underneath that ice layer, so that that ice layer protects the plants, insulates the plant, and protects from the severe harsh winter breezes and all. So that's what they use as strategies. But still, like in early springs or early winters where they cannot make enough ice, they do face severe problems. All these cranberries face severe problems. That is the reason we want to study like cold tolerance gene and to improve cold tolerance, cold hardiness in cranberry. So technically, like I am studying two types of cold tolerance genes here. One is anti-freeze proteins. We also call them as ice binding proteins. These ones modifies the growth of ice crystals on the membrane. You can see like this is the ice crystal. These anti-freeze proteins, they change the growth pattern so that it won't damage the membrane at all. So, and I am also studying dehydration responsive elements. These dehydration responsive elements also helpful in drought and heat stresses too. They technically help to withstand dehydration due to extracellular ice. So, and we measure like gene expression of these in buds and leaves during winter, and compare them when there is no cold. You can see like this one is technically there is no cold. So you can see like all genes were upregulated. So in buds, we see most upregulation of the dehydrins, and in the leaves, we found cold binding factors upregulation. So every organism has different mechanism. Every tissue, leaf tissue has one different mechanism, and bud has different mechanisms. So we are still working on it. So the summary of this transcriptomics is we found significant differences in gene expression. So our next step, and we are in the process of isolating, cloning, and identifying the sequences of these genes. And then our next step is to overexpress them, or using CRISPR technology, silence them and study the gene function of this particular cold tolerance genes, and use these genes as a markers for screening and breeding for cold hardy cultivars. So another project I am working on is metagenomics, that is also omics, to identify the cranberry microbiome. Microbiome means basically the beneficial bacteria and fungal and archaea available in the soil and the plant, cranberry plant. Usually these promote plant growth and they also help with plant immunity and they influence the soil structures. So we are using metagenomics to sequence these bacteria and fungi and other microbes in the soil, cranberry soils, roots, and aerial parts. So our goal is to identify, isolate them, and see how they are useful to the plant for nutrient uptake and immunity. So once they are beneficial, once we identify the beneficial one, we want to recommend them as a biofertilizers. So this is just like we have huge amount of data from this microbiome. I'm just showing like 3 microbes. So we collected samples from conventional ones, conventional soils, organic soils, and wild cranberry soils, and wild soils where there are no cranberries but other ericaceous species like blueberry, lingonberry, and different ones. You can see like compared with the conventional or organic, this wild one has huge amounts of bacteria. So and also we found mycorrhiza also. Um, Mycorrhizae is a beneficial fungi which helps in nutrient absorption. And also we are trying to identify the signaling molecules between bacteria and mycorrhizae to see which one collaborate with each other, which one symbiotically work with each other so that we can recommend that to growers. So, and from these wild organic and conventional soils, we isolated different bacteria. First one here is phosphorus solubilizing bacteria. So basically we add this, this blue marker here, and this phosphorus solubilizing bacteria, it solubilizes all. So you can see clearly it used all and made the plate clear. So this phosphorus solubilizing bacteria solubilizes phosphorus in the soils because phosphorus is not as movable as other elements. So and makes it available to the plant. So and also we isolated some ammonium fixing bacteria. Cranberry uptakes nitrogen in the form of ammonium, not in the form of nitrate. So we found this ammonium fixing bacteria from soils and the roots both. So that they can be helpful in fixing ammonium instead of adding synthetic fertilizers. So and also we isolated some auxin promoting bacteria from the roots of cranberries so that they can helpful in promoting root growth. So with this, as I said, like we also have project going on side by side with mycorrhizae, not only bacteria. So with the time limitation, I don't want to show everything here. So we want to identify them, isolate them, and then try them with the cranberries in the greenhouse and also in the field conditions and see which one is helping more for the uptake of the nutrients and the immunity. And then we want to recommend them as biofertilizers to the growers. So overall summary of the inomics. So these cranberries are unique because I work with different crops. I didn't see like the management like they do for cranberries. It is entirely unique and different. So it's awesome also because of its all medicinal properties and how people eat for vitamin C. So when during COVID time, most of the people bought cranberry juices. So because it has a lot of vitamin C. So we are using different omic technology to understand molecular and physiological mechanisms to improve its tolerance to abiotic and biotic stresses. So next one I want to talk about is goji berry, how and their omics. This is a brief study. I did a lot with goji berries, but I want to present only a little here. So goji berries also called as wolfberries, and they belong to the family Solanaceae, and they usually cultivated in Asia and mostly China. I have collaborators in China that are working on goji berries. So they are mostly used as herbal trees and dried berries. They are rich in polysaccharides, antioxidant, and vitamins. So it has health benefits like anti-aging, anti-cancer, diabetes, and high blood pressure, improving immunity. I have seen most of the Chinese people like they add dry goji berries in the hot water and they drink for anti-aging actually. So I studied mostly these goji berries responses to elevated CO2. As you can see, like our CO2 is increasing over the years. So now the ambient CO2 levels are 400 ppm. So, and also on this figure, you can see like as the CO2 increases, um, so the net photosynthesis increases. However, we compared here like C3 and C4 plants. C4 plants have optimization for photosynthesis. Now they are saturated. So even though if you provide more CO2 concentration, it's not going to be useful for C4 plants. But C3, they do improve their photosynthesis with the CO2 concentration, but at certain level, they become plateau. So because goji berry is a C3 plant, we want to see how this increase in CO2 levels, our hypothesis is that increase in CO2 levels will improve sugar levels in the fruit. So, and we used open top chambers, you can see, where you can control temperature and CO2, and we grew them in there, providing a lot of CO2, and also we grew them outside for ambient CO2 conditions. Our assumption was that they do improve total sugars and everything, but we measured these sugars, flavonoids, and carotenoids, and also in total sugars, we measured different sugars, thinking that they will improve, but they did improve until certain stage. But when the fruits are ripened, we could clearly notice decrease in total sugar content. Um, this is totally opposite to our hypothesis than ambient CO2 conditions. However, they do improve their flavonoid content. And also carotenoids in the goji berries were also reduced due to elevated CO2. So we want to find out the reason why they drop the sugar levels, even though they are C3 plants. One might be photosynthesis acclimation, or maybe some genes are responsible. So here we studied like transcriptome profiling, transcriptomics, where we studied gene expressions. So we found total 73 genes differentially regulated between ambient and elevated CO2 levels. Some of them are upregulated and some of them are downregulated. So out of these genes, we identified three genes, three sugar metabolism genes. Those are UDP glucuronate and malate synthase alpha galactosidase. These three genes are involved in sugar metabolism pathway, 4 sugar metabolism pathway. Uh, so these are like kind of main, like they, I didn't present, like I'm not showing the figure here. They are everywhere in the sugar metabolism pathway. So these are differentially regulated to elevated CO2. So that's why we isolated them, we cloned them, and we sequenced them. We identified the protein structure of these three genes. And also we overexpressed them in tomato and studied like their functional analysis and everything, whether they are really responsible for composition of or reduction of some sugars under elevated CO2. Here I am showing only one. So this is alpha galactosidase. Here in this family, we identified 7 sugars in goji berries. And then we isolated them and we sequenced them and we compared the sequences. and we measured their expression levels to ambient and elevated CO2 and at different growth stages. And you can see like in different fruit stages also. And then we overexpressed them in tomato and studied how they respond to the elevated CO2, increase in CO2 levels. And we measured relative expression, and also we measured galactosidase enzyme activity. So you can see under elevated CO2, it all increased. So all galactose increased and sucrose increased and fructose increased, but glucose and polysaccharide levels decreased. These polysaccharide levels are very important for goji berry because they give most medicinal properties. So we identified some overall, like with the goji berry study, we did identify sugar metabolism genes that are responsible for different composition and reduction in the sugars under like elevated CO2 levels. So overall summary for the goji berry is goji berry do respond to climate change by reducing sugars and polysaccharides in mature fruit. And we studied sugar regulating mechanism and identified genes that respond to elevated CO2. And we isolated and sequenced these genes so that we can use this information to produce high quality goji berries under future climate change conditions. So that's all I want to present. So here my team, I want to thank all my team members for their hard work and doing this much work in 2 years. So, and also I would like to thank all my cranberry growers who helped me to understand the crop and who provided all the support. So thank you very much. Thank you for the invitations once again, and thank you for the opportunity. So please go ahead and ask like if you have any questions.
Thank you, madam. Thank you very much. Excellent presentation of cranberry and gooseberry. And I have covered cranberry. It is a Thanksgiving meal, excellent, and it has a lot of medicinal properties and rich in vitamin C, and which leads to immunity. And I have covered so many value-added products of cranberries, and 70 varieties are in cranberries. You have covered by topic omics, genomics, artificial intelligence methods to optimize inputs, and growing degradase, ionomics, impact of pH and fruit size, foliar sprays, transcriptomics, metagenomics, genomics, and biofertilizers. And I have covered everything in gene transfer and how they are responding to different biotic and abiotic stresses. And gooseberry, it is a herbal tree plant, medicinal properties, and I have covered fruit sugar metabolism pathways, and response of gooseberry to elevated carbon dioxide. And excellent presentation. Thank you very much, Dr. Jyotsna. And now it is open for the discussion and the question and answers. Madam, Dean Madam, and all university officers, followed by zonal heads, associate deans, and students, scientists.
Thank you, Dhananjay Garu, and thank you, Dr. Jyotsna. It's really very, very informative and very elaborate discussion regarding the physiological and molecular aspects of cranberry. In fact, let me be frank, cranberry, we are not very much aware of the details of this cranberry because it's not our South Indian crop. Hardly we have studied about this crop because in our BSc or MSc horticulture, we have concentrated mostly on the South Indian fruits and giving less importance to other fruits. Really, we have given very good information about cranberry. Certainly, it will benefit. The information is definitely benefit our students and staff. Regarding the molecular and physiological aspects, really detailed information you have given about these aspects. And think because is there any possibility of growing this cranberry in our region? What is the minimum
No.
temperature requirement for its growth? Because you told organic soils and mostly acidic soils are suitable for it. So in in southern parts of India, there are certain cooler areas. Is there any possibility to grow some heat tolerant types?
Actually, there are some cranberries that are available in southern United States too. So, but they do need chilling requirements for dormancy. So I believe like we don't have those requirements in India. So not only southern part of India, I don't think so. We can grow them in northern India also. But all this information is also useful for developing blueberries. So I'm not only working on cranberries, but I'm working side by side on blueberries. We can always translate these information to other fruit crops. So because it's not only like all these identification of abiotic or biotic stress tolerance genes or identifying biofertilizers, not only useful for only cranberry, they can be useful to the other crops too, other fruit crops too. So that's the reason we are working on it. So all this information, I'm pretty sure, like the crops, whatever we are growing, fruit crops we are growing in southern part of India. So we can translate the knowledge to those crops.
Okay, ma'am. Thank you very much. Thank you for the nice information. And I think the day has already turned there.
So thank you all for giving the opportunity. You are always welcome. I know like it's not a crop, desirable crop in India. So, but yeah, like if you need any information or anything, so anybody, all students or everybody can contact me. I don't know whether I provided my email or not, but
You have given an email, madam. Will give, no problem. And any university officer, Dr. Karunasri, Dr. Salami Sunita.
Good evening, madam. Dr. Josna.
good evening. It's like morning only to me.
Okay. Madam, last year we got just introduction about this crop from one of the Angro alumni. Like she was presenting about through bioengine series. We got just introduced about this crop. Yeah. Leela Sai Sri Uppala.
Yeah, she is my friend.
Okay. Like she was telling about some biotic stresses like that. What is the status of any biotic stresses in your area, madam?
So I am working collaboratively with Leela Uppala. Actually, we both are working on this microbiome project. That's our combined project. So we want to, along with beneficial microbiome, we also want to see what is causing fruit rot. She is specifically working on fruit rot. So fruit rot diseases, are there any bacteria that can suppress this fruit rot fungi? So we are still working on it actually, because we both started recently working on cranberries. So it takes, I believe, like 2 more years to completely identify some bacteria that can suppress fungal diseases. So
what are the major diseases in your area, madam?
Here for cranberry, I can say fruit rot is the major disease, because it cannot tolerate even small heat stress. So that causes, scald on the fruit, and then that causes attack of the pathogen. Not only heat, like, even if you have a lot of humidity, that also causes like producing a lot of fungi at the canopy level, and that damages the fruit. So
is it Oomycetes fungi anything, madam? Is it Oomycetes fungi or what are the exactly etiological agents?
There are, I'm not an expert in fungal diseases.
No, I'm just inquiring like.
There are 20, I think, dozen fruit rot fungi. I don't know exactly. I am working on molecular side, identifying without, culturing those bacteria, and without looking underneath the microscope. my work with Leela is to, developing the markers and then isolate the DNA and use the markers and tell which fungal disease it is. That's what I'm helping her with. Um, so, yeah, sorry, I cannot answer
Yeah.
that question.
No issue, madam. Like I was thinking, like because you were telling about low temperatures, probably this Pythium phytophthora, those Womystis fungi may be predominant.
Phytophthora is there. Phytophthora is the main reason for fruit rot fungi here. So it is one of the fungi actually.
Yeah, probably it
That
might be a
causes-
complex disease. So many etiological agents might be there.
Yeah, there are 12 actually. They identified 12. We are trying to identify more with the microbiome analysis. That gives like whole sequence data, right? So what is in there exactly? So that's the reason we are using molecular tools instead of like, culturing and all. So maybe we will come up with more fungal factors there. So not only the one identified now. So
It was very nice listening to you, like applying all the latest technologies, genomics, transcriptomics, artificial intelligence. It is very nice. It is very inspiring, madam. Congratulations.
thank you very much. Thanks. Appreciate it.
Seshakaran, thank you. Next, anybody? From all that any scientists?
So can I make an announcement here? I don't know how to do the messaging because I'm using web browser. So I have a friend here, Sirisha. I will contact you. So
Joshna, nice to see you. Okay, later on you can message.
yeah, I will contact you. So it's been so long.
You want to ask anything, Dr. Josna?
No, I will contact her. I have her contact.
Okay.
Yeah.
If nobody is there, we will go for the vote of thanks by Dr. Salami Sunita, Dean of Student Affairs, Dr. YSR Horticulture University.
Yeah, thank you very much. And please email me if you have any questions.
Sure.
It's my pleasure and privilege to propose formal vote of thanks to this Global Connect series of today's program, that is Series 8. So my sincere thanks to Dr. T. Janakiram sir, Honorable Vice Chancellor, Dr. YSR Horticultural University for facilitating such a meaningful and novel program of international exposure to all the 42 institutions of Dr. YSRHU. Thank you very much, sir. And this program is helping us a lot to have an international research exposure. Thank you, sir. And my heartfelt thanks and congratulations to today's speaker, Josna Devi Mura. Really, we are much thankful to you for your excellent presentation with such a clarity. So it is like listening to a story of cranberry sitting and you like from A to Z of the crop you have explained to us very clearly, and how the crop has come to existence or history and harvest, everything. It is so interesting and informative, and there is much clarity in your presentation. And from simple things like phenomics and the development of camera, that is the technology development, it is a great encouragement to youngsters and the students, PhD students, and the way you are organizing your research. And in metabolomics, you have a long journey to go ahead, and everything so elaborately and very clearly you have explained, madam. And like ionomics, antifreeze proteins, and coming to the goji berry also, how you have worked with the sugar metabolism, the genomics part of it. So very excellent presentation. Thank you so much. And you have a great journey ahead in your research career. And congratulations for that. And my sincere thanks to all the university officers, Dean of Horticulture, Dr. A.S. Padmavatama Madam, Dean PG Studies, Dr. S. Suryakumari Madam, Director of Research, Dr. L. Narayanaidu Sir, Controller of Examinations, Dr. D.V. Swami Sir, and Director of Extension, Dr. E. Karunasri Madam, for your active participation and interaction during the session. And I'm thankful to all the associate deans, heads of the research stations, and heads of all the 42 stations or institutions of Dr. YSRHU, all the senior faculty members, teachers, scientists, and students for your active participation in this program. And I'm thankful to the rapporteurs, Dr. V. Sudhavani, Associate Professor and Head of Post-Harvest Research Station, College of Horticulture, Venkatramanagudam, and Dr. C.P. Vijay, Associate Professor, Entomology, College of Horticulture, Venkatramanagudam. And my sincere thanks and congratulations to the convener of this program, Dr. K. Dhanunjay Rao Sir, Director of Industrial and International Program, Dr. YSR Horticultural University, for planning and executing the program in a successful manner. And thanks to the non-teaching staff of the section also. Thank you one and all for a wonderful time. Thank you so much.
Thank you, Madam, Dr. Salami Sunita. And thank you one and all. And Dr. Josna Devi Mura, thank you very much. And now it's 12:30 midnight, I think. We are sorry disturbing you.
No problem. I'm happy to actually present anytime. So thank you for the opportunity, everyone. So there are so many people. So
Yeah.
appreciate all your encouragement and support. So please feel free to contact me. So I kind of, I don't know whether I provided my email or not, but I will provide. So yeah. Thank you all.
Thank you, madam. Thank you, one and all. Definitely we will be in touch with you in the future also. And thank you very much. Now it is closed. Thank you.